So in this video, we're going to be talking about diffusion and osmosis. So plasma membranes, as we talked about, are selectively permeable. So small nonpolar molecules go through the plasma membrane very quickly, followed by water, followed by glucose, right? These are both polar, but water is smaller than glucose. And then ions, which are electrically charged, don't go through the lipid portion of the plasma membrane really at all. And so in this video, we're going to talk about how these small molecules move through the plasma membrane. So there are two types of movement across membranes. There's passive transport and active transport. And passive transport occurs when a substance diffuses across a membrane in the absence of an energy source. Okay? This is in contrast to active transport, and we'll talk about active transport in the next class, and that requires energy. And there's two types of passive transport. There's diffusion and osmosis, it's one type, and there's facilitated passive transport. And this type of transport occurs through proteins. And we're going to cover this also in the next class, but today we're really going to focus on diffusion and osmosis. So let's start by talking about diffusion. So first, let's define what a solute is. Small molecules and ions in a solution are called solutes, and they have thermal energy and they're in constant motion. And the spontaneous movement of these solutes is called diffusion. A concentration gradient is created by a difference in solute concentrations. And when a concentration gradient exists, there's a net movement from high concentration to a region of low concentration. Okay, so let's talk about this in a little more detail. So let's say a bunch of people are standing in a room packed tight in a circle in the center of the room. And they're all somewhat moving and jostling. They all have thermal energy, right? And they're going to randomly bump into each other, right? And keep bumping into each other until they've dispersed, okay? So there's a net movement, right, from areas of high concentration to regions of low concentration when a concentration gradient exists. And diffusion along a concentration gradient, it increases disorder, right? Because now instead of standing all in the center of the room, really organized, you're dispersed all over the room. And this increases disorder, and because it increases disorder, this kind of movement is spontaneous. It doesn't require any energy. And equilibrium occurs when all the molecules are randomly distributed. And so everyone's still kind of milling about and moving randomly, but there's no net movement from a concentrated area to a less concentrated area. And that's equilibrium. And so that's shown here. Okay, so we've got our planar bilayer experimental system here, right, where we've got a flask, and in the center of this flask there's a lipid bilayer. Okay, we've got solute purple triangle here, and you can see that this solute purple triangle is really concentrated on this side of the lipid bilayer. And so it's going to diffuse from areas of a high concentration to an area of low concentration until it's in equilibrium. Now, solute white circle is very high on this side of the lipid bilayer, and so it's going to diffuse from an area of high concentration to an area of low concentration. Right? So here's our concentration gradient, here's the really high, we're really concentrated, and this solute is going to move from an area of high concentration to an area of low concentration. And this is going to happen spontaneously if the solute can cross the plasma membrane or the lipid bilayer. The second example I want to talk about is the process of osmosis, and this is the special diffusion of water. Okay, so water moves quickly across a lipid bilayer, and this special case of diffusion is called osmosis. And it only occurs across selectively permeable membranes. So if a solute cannot cross the membrane, water will move across the membrane. So water moves from areas of low solute concentration to regions of high solute concentration. Okay, so this is a little bit opposite in a way than how solutes move. Okay, so what's happening is that if a solute is really high on this side, or let's say we've got really salty water over here and really diluted water over here, salted, right? But the salt can't move. Let's say that it's a bunch of ions. So if those ions can't move, water then moves from that low salt concentration into the high salt concentration, thereby diluting the concentration of the solutes so that it equalizes the two concentrations on both sides of the bilayer. And so that's shown here, okay? So here we have our planar bilayer, and here we have low solute concentration, and here we have high solute concentration. But let's say these solutes can't cross this lipid bilayer. What happens then is water moves by osmosis from this area of low solute concentration to this area of high solute concentration, thereby equalizing the concentration of solutes on both sides. Okay, and so this is also shown here in this jelly bean, what are these called? Oh, this is shown here in this gummy bear experiment. So if you ever need to show children how osmosis works, this is a great experiment. And so what you do is you take these gummy bears and you dunk them in a glass of water. And then you wait overnight and you pull the gummy bears out and they're all plump like this. And that's because, right, here we have these gummy bears that are fairly high solute concentration. There's a ton of glucose in these gummy bears. And so the water on the outside is going to move from low solute concentration to areas of high concentration, and they're going to make these gummy bears swell. And so, right, that's the net movement of water by osmosis. So there's a couple different ways in which water can move that depend on the type of solution that the cell is in, okay? And those three types of solutions are characterized as hypertonic, hypotonic, and isotonic. And we're going to talk about each one of those solutions and what happens to the cell when it's in those solutions, okay? So the first example we're going to talk about is a hypertonic solution. And a hypertonic solution occurs when the solute concentration is really high on the outside of the cell here versus the inside. Okay, so this is an example when you place a cell, for example, in really salty water. Okay, so there's a lot of salt on the outside. And because there's high salt on the outside, the net movement of water is from areas of low solute concentration to high solute concentration. And because water is leaving the cell shrivels up and shrinks. Okay, and the way I remember hyper is if your kid is hyper, throw them outside to play. So hyper meaning that the salt is all on the outside and the water then leaves the inside of the house to go outside to play, leaving the cell all shriveled up. The other instance is a hypotonic solution. Okay, so a hypotonic solution is when the solute concentration is low on the outside of cells and high on the inside. Okay, so the water will move from areas of low solute concentration to high solute concentration. So now all the water is flooding inside the cell, and then the cell will swell or even burst. And when a cell bursts, we call this lysis. And so here we have a cell bursting because we put it in a hypotonic solution. And this is an example of if you put a cell in pure water. So occasionally we make this accident in a lab where we mean to put our cell in a buffer that has a nice happy salt concentration, but instead we drop the cell in pure water and then we've ended up lysing all of our cells. And the last example I want to talk about is an isotonic solution. And so this is when the salt concentration is the same on the inside of the cell as the solution it's in. And when this happens, water moves evenly across the membranes, but there's no net movement. And so the cell is happy and there's no change in its shape or size. And this occurs really in around 0.9% salt or 0.9% saline that's been diluted in water. This is a really nice concentration of salt for a cell to be in. So in summary, diffusion of a molecule occurs down its concentration gradient. This movement happens spontaneously because it increases entropy. The plasma membrane is selectively permeable, right, in the order of small nonpolar to small polar to large polar to ions. And osmosis is the movement of water from areas of low solute concentrations to area of high solute concentrations. And the movement of water in a cell can depend on whether the solution the cell is in is hypertonic, hypotonic, or isotonic. And we'll talk more about these in class, as well as examples of diffusion. And so that's all I have to say today, and I look forward to seeing everyone in class. Thanks, bye.